How's it going, everyone? Okay. All right. So I'm from LBNL. Work with Chris Bungel's group and others there. All right. So let's get started. I actually have this maybe surprising result to me, at least. I've worked with microbes for a long time. So these are estimates of costs and benefits that we get as a society for microbes, right? There's some very interesting things here. One positive is we've probably already reached like $1 trillion bioeconomy, which maybe not everyone knows about. So that's on the left. And then on the right, we have the costs. So the costs, of course, are much, much larger. They're partly much larger because of human health, right? So antimicrobial resistance, maybe biosecurity, right? Also animal health, agriculture, plant health, food safety, lots of important things there. So there's a really big mismatch, right, between the costs and the benefits. And one idea is that if you can learn about microbes, right, and their growth preferences, you maybe have some very easy ways to try to mitigate this, right? Maybe increasing salt concentration or pH, et cetera. One other piece maybe is that the market value for the cost is much lower, right? So somehow we're not accessing or tapping into the capabilities there. Okay, so we realize that cultivating microbes is a bottleneck. There's lots of different microbes. They're getting discovered daily, more or less, but in diverse environments. And culturing is a starting point. There are other issues in terms of cultivation, like slow culturing or reproducibility, and just the expense and price, right? But there exists a lot of prior knowledge, mostly in literature, about these different growth preferences, things like temperature, oxygen, different kinds of traits. It's very fragmented. This literature goes back maybe 200 years from different countries, right? Different styles of analysis, research, a lot of unaligned namespaces. And because of all this, we can't really deploy this for AI ML, right? Okay, so I think here, everyone's familiar more or less, right? That there exist technologies out there that recommend movies for us, recipes, food, based on prior data, right? And ontologies and knowledge graphs are a pretty key component here, right? They help standardize information. They provide ways to make relationships between things. And then they plug into a whole family of AI techniques that can devise recipes or suggest pairings, right? So these recommendation engines. And one subfamily of these methods is graph learning and link prediction, right? However, to do this, it requires a knowledge base, in this case, of existing recipes, right? Or growth preferences, other data about microbes. And in fact, we still don't know which pieces of information are going to be the most valuable. So we need to do some discovery exploration here. So this project started as a prototype component for self-driving labs. That's kind of the proposal. That piece is maybe still some ways away. But we're kind of trying to prove that you can do learning and feed data back into a system of knowledge. So we start with constructing a knowledge graph. We train models, make predictions. Those get validated experimentally. And then the data from those experiments can be fed back into both the knowledge graph itself, right? And then into updated models. And one key piece here is the negative data, right? Negative data probably is the bulk of scientific efforts out there, but it's just so inaccessible, right? It doesn't get published, doesn't get released as data sets. And so here, because we have a close collaboration with experimentalists, we can capture that, right? Model it and put it back in and use it. Okay, so KG Microbes started about 4 years ago. It's actually an offshoot of a COVID project, KG COVID-19, if you're not familiar. So we're using some of those technologies. In maroon here, I'm marking all the data sets that we've already modeled and ingested. There's a kind of cloud of extra things there. We focus on traits and growth preferences. And I think we've touched all of the major resources for this that have, at least 10,000 or so taxa. So there's Mann et al. Publication, Back-to traits, and of course, Backtime. The other data will probably involve some new techniques to process quantitative information into semantically usable things, right? Like growth curves, for example. And we've also been lucky to co-develop some new text mining methods using LLM assistance, right? So here you can use something like a structured template approach and give abstracts, right, or other information and pull out properties of microbes in a standardized fashion. To support this work, we had to actually develop an ontology. So this is available on BioPortal now. This focuses, again, on these growth preferences and categorical traits. It uses a robot template So there's a Google sheet that anyone can really interact with, even if they're not computationally savvy, experimentalists or microbiologist experts. So that's very handy. And we have an ODK workflow to support this and releases on GitHub. But an important piece here that we're trying to include is to cover both categorical terms like aerobe, anaerobe, and then the quantitative versions of those, right? So you can think of temperature as some range. You can bend that range. And now you have terms that correspond to parts of that range that then allow you to extract quantitative data from publications. We took this modular KG construction approach. That really is kind of the spirit of KG Hub and other pieces of tooling that we use. This was with our great collaborators, Brooke Santangelo, Kathy Lizupani, Larry Hunter from CU Anschutz. KG Microbe Core was kind of this original KG, right, that we built based on established ontologies and these trait resources that I mentioned. Onto this, we can layer other data, right? So there's a biomedical version that includes host phenotypes and diseases and some dysbiome information. And you can imagine doing that for other hosts, like a plant, for example. Now the function data, function annotations are much larger. They put you into different scale. So we separate those, right? We have separate KGs that include microbial genome annotations from UniProt. Still a bit limited. There's so many microbes, right, that will be going on for some time. But we have some extra tooling to operate on these and could do queries and other pieces here. And at the bottom here, there's a bunch of citations for various aspects here. Okay, so on to semantic modeling. This is what the KG microbe graph represents. It has a hub for organisms, lots of relations to different types of traits and phenotypes, things about antibiotic resistance and sensitivity, cell shapes, lots of flavors of growth preferences, pathogenicity, et cetera. We also can model assays here. Let's see if I can point. Maybe not, but there's a little assay node under organism, and we can contextualize those assays by linking them to nodes for media and various parameters of an experiment, right? So that's maybe something a bit new. There's another hub on the right that are based on proteins. Those are the genome annotations. Those live as ECCB, REA, Go terms directly from UniProt that we trust. They're probably somewhat incomplete, right? And not that many genomes in the end. But it's a good data set. And then to support all this, we also have the full REA reaction network. So you can do walking in reaction space. And of course, that also includes pathways. All this is available on our GitHub repo, including these data products that you can download with hundreds of millions of edges, for example. And now on the left here, we have a subgraph for a very important industrial organism that is used to make amino acids, Corynebacterium glutamicum. And in this layout, you can see that in red, there are lots of related strains to it, right? It's a high value organism. So people have sequenced it and edited it, right, in various ways. The other circles in there are things like isolation sources, chemical assay results, and these growth preference traits. So it gives you a view, right, of a little subgraph for a specific organism. And we have about 100,000 of these. This is an analysis that Brooke did for our paper, a preprint that you can check out. And it's an important example because it takes definitions of pathways based on reactions from this Vital et al. 2018 paper, 4 different ways of making butyrate. And essentially, if you satisfy these reaction equations, then you're predicted to be capable of this pathway. But in the KG, we can overlap this information with assay results from BACDIVE, for example. And so here, there are a few different analyses that show you that maybe not all things agree and that you still have limited information, right? But you can still find agreement between a genome annotation and a true assay result. Okay, so why KG is good for AIML? This may be surprising or not. A couple layers to this, and actually there's maybe 1 or 2 more I'm not showing here. We can maintain links to the raw data. So here, because of the KG Hub and COSA, other types of tooling, we can go back and look at text information, for example. Mostly we use the graph adjacency matrix, so this binary matrix, as well as some graph paths and graph embeddings. This was a collaboration with our great collaborators for graph learning. Okay, so we're able to train about 45 models for the most common media using CatBoost for fast experiments. We then made predictions for over 20,000 taxa. These didn't have media associations in our KG, right? So we didn't know the true answer. And then here we have some examples on the bottom about, about the top 5 predictions. And you can see that there's some overlap between the species names and the media, right? So we have some initial clues that things start to make sense. There are different ways of training these models. They can be more or less expensive. And really, we're interested in experimenting with different types of features, right? So that led us to this result, which I think is surprising and not surprising. So first, the best models have about 70% precision across these 45 media. There's kind of a distribution of performance there. They have fairly low recall, right? So you're still missing a lot of taxa. It's actually amazing that these sparse traits give you that much information, right? So this power in things like cell shape and growth preference above and beyond what we can annotate in the genome. So that legacy information in these papers is still useful. Let's not forget about it. But maybe another surprising thing here were that just the enzyme and reaction annotations, right, actually give you a fairly poorly performing model. So I think this is just a genome annotation gap, right? We spend so much effort on sequencing, storing sequences, and maybe function classifications, right? But they're really not touching these important things that are emerging out of maybe interactions between multiple genes and other, right, more complex relationships. Now in this result, we include eggnog mapper. So eggnog mapper is kind of a de novo genome annotator. Lots of those features look like, this sequence family is conserved in clade X, and that's all we know about it. That alone, starts approaching these complex traits, right? But because it's unexplainable, it's kind of a limited use and probably would involve a lot of experiments to make anything useful out of it, right? You could probably do knockouts of those genes, see what happens. Could be a long road. Probably we should do those experiments. Okay, so just for sake of time, I'll keep going. We've tried a couple other approaches here for predictions. One is symbolic rule mining. So this is with our collaborators in VSC Prague, using this clever miner tool. And we like these, and biologists love these, because it essentially gives you a sentence with a statistic, right, for predictions that you could actually, a human could edit, right? So these rules, we're essentially finding a set of rules, stringing them together into a classifier. And for now, we have preprint on this. I would say this is still a fairly simplistic approach. The rules, have 2 terms on the left, right? And you can essentially walk through that list of rules and see which ones matches your case, right? And then you get a prediction. You can get these rules to be much more complex. So here on the bottom right is a rule that's mined directly from the RDF structure of the KG. That's kind of like a Horn rule, like a first logic version, I guess. And here we're predicting growth in a media based on presence of an enzyme and also linking consumption production of a specific metabolite. So I think this will be an exciting direction, kind of more in this neurosymbolic space. And again, this beta-galactosidase is an enzyme that's important for breaking down complex carbohydrates like agarose or carrageenan, stuff that they put into our processed ice cream and yogurt, for example. So also useful for our microbiome, right? But these are, in the marine world, these are actually real biological structures that exist, right? And so microbes like to eat that. The rules and features can be complicated to explain. So here we use LLM enhancement. We make this fairly complicated interaction with LLM. This is at the bottom here. So it includes a prompt, it includes a recipe for the media. It includes complex ingredients that we also source with an LLM with citations. So these are all with deep research, where you can click on a link and check that that is the paper, right? And that it makes sense. And this was actually fairly fruitful. So here I'm showing you 2 examples of 2 sets of features that are explained by an LLM to have some association with specific media components. So the first one are these different marine isolation sources, just different hosts or organisms that live in those environments. And here you can see that, to recreate that seawater environment, you need a bunch of different kinds of salts, right? And appropriate concentrations, and this gives you that linkage to this environment. The bottom row is actually a little more subtle, right? This is about gram-stain negative, so property of the outer membrane of these microbes, where the liposaccharides require specific cations to be functional, right? And those are present in this media. And there's kind of more examples of this that you can go through. In addition to media, we also predict models for growth, physical growth preferences. So in this case, temperature. And I'm showing you this plot, which is actually, this model is trained just on those ECCB reactions. In this case, it works fairly well for the multiple temperature categories. But the SHAP plots have this kind of amazing volcano plot property where there's a good separation between positive and negative effects, right? And when you compare the extremes of temperature, so psychrophile on the right, hyperthermophile on the left, and you can repeat this for other types of properties, right? You actually get a fairly explainable list of features. Something that maybe wasn't known is that catalase is essentially only present in psychrophiles, probably related to higher oxygen concentrations at low temperature, right? Whereas these RNA stability functions are present in hyperthermophiles because RNA doesn't, is less stable at high temperature. And there are kind of more cases of this. Okay, so I'm going to speed up here. My last 3 applications. You can do also neurosymbolic reasoning using these KG embeddings, right? So we have millions of nodes and embedding for each node. And we've probably seen these analogy reasoning cases, right? I don't know what the equivalent of Liverpool is, but I do know what the equivalent of Akkermansia mucinophilia is. This is our gut microbe that likes to eat our mucus, probably a very promising probiotic, but it's an anaerobe, very difficult to work with, right? So our collaborators came and wanted us to find the closest microbe in trait space, that's an aerobe. And so essentially you can perform this vector analogy reasoning equation. And now the top match to that is an aerobe from acidic hotspots. So it matches our acidic environment in our gut and the higher host temperature. All right, just quickly 1 or 2 more. So we're also recently been able to train relation graph transformers. And for now, the result I'll share with you is we can cluster these relation embeddings, right? So these are all the types of relationships in KG microbe. And what we're getting out of this is these fairly clean groups of types of relations that separate, biological process from structural relationship, from causal from functional. And at least this provides us a very good QC aspect for these very large data products, right? They're kind of hard to look at. TISNE really doesn't do it justice, right? All right, and just to end, very recently, we've now deployed these onto the GPT tool with the template to extract information for text. So we're getting triples grounded in ontologies, including METPO and taxa. These are just 2 example statements, right? And there's still some issues with taxonomy, so strain level may be difficult. In some cases, you don't have the sequence layer here, but I think a lot of that can be addressed soon. So we're going to working on running this for 80,000 papers. Okay, so just maybe I'll leave you with this slide. We'd love to collaborate, so please contact us. And I just wanted to acknowledge all the great people involved in this work. Harshad was kind of the main developer for a few years. Brick is the first author on our paper. And then Mark and Sujai have been helping more recently with ontology and the text extraction.
Fantastic, thank you. We have time for 1 question.
You made a pretty big point about how a lot of the work is benefits from your experimental collaboration and the lack of negative data in the microbe space. Are you guys releasing the negative data from your experimental results?
Oh, will we release it?
Yes.
Yes, of course. Yeah, so we'll actually put it in the KG once we get it, hopefully in any week now, and we can model it specifically as, for example, it does not grow in, right, or does not like this nutrient. And that'll be data that's, exactly in the KG, hopefully setting some precedent for people being able to share that now, right? Because that has been a challenge, right?
And the answer to the Liverpool of America is Philadelphia, for sure.
Philadelphia. I was going to say Seattle, yeah, right? But no, good. I thought of Philadelphia too, right? Cheers.
Awesome. Thank you very much.